Have you ever pondered the importance of translation in protein synthesis? Well, it's as crucial to life as the assembly line is to a car factory. But instead of producing cars, this biological process manufactures proteins, the building blocks of life. Let's dive into the process of translation, a term in biology that refers to the polymerization of amino acids to form a polypeptide. The sequence of these amino acids is determined by the sequence of bases in the mRNA or messenger RNA. These amino acids are linked together by a bond known as a peptide bond. This bond formation requires energy. Thus, in the first phase, amino acids are activated in the presence of ATP and linked to their corresponding tRNA. This process is known as charging of tRNA, or more specifically, aminoacylation of tRNA. Think of this as a matchmaking process where each amino acid is paired with its corresponding tRNA. If these charged pairs are brought close enough, the formation of a peptide bond between them is energetically favored. The cellular factory responsible for synthesizing proteins is the ribosome. It consists of structural RNAs and about 80 different proteins. Ribosome exists as two subunits in its inactive state, a large subunit and a small subunit. When the small subunit encounters an mRNA, the translation process begins. The large subunit of the ribosome has two sites where subsequent amino acids can bind to and be close enough for the formation of a peptide bond. Ribosome also acts as a catalyst, enhancing the rate of peptide bond formation. A translational unit in mRNA is the sequence of RNA flanked by the start codon, typically AUG, and the stop codon. This sequence codes for a polypeptide. mRNA also has additional sequences that aren't translated, referred to as untranslated regions or UTRs. These UTRs are present at both ends of the mRNA and are required for an efficient translation process. The ribosome binds to the mRNA at the start codon, recognized only by the initiator tRNA, marking the initiation of translation. During the elongation phase of protein synthesis, complexes composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA anticodon. The ribosome moves from codon to codon along the mRNA, adding amino acids one by one. These are translated into polypeptide sequences dictated by DNA and represented by mRNA. Eventually, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. So, in essence, the process of translation in protein synthesis is a meticulously orchestrated dance of molecules, where mRNA, tRNA, and ribosomes come together to produce proteins, the fundamental building blocks of life. It's a testament to the intricate and marvelous nature of biology, reminding us that even the most minute processes within our cells play monumental roles in the grand scheme of life.